Phytoremediation, meaning plant, and Latin remedium, meaning restoring balance, describes the treatment of environmental problems through the use of plants that mitigate the environmental problem without the need to excavate the contaminant material and dispose of it elsewhere. Phytoremediation is a cost-effective plant-based approach to remediation that takes advantage of the ability of plants to concentrate elements and compounds from the environment and to metabolize various molecules in their tissues. Toxic heavy metals and organic pollutants are the major targets for phytoremediation. Knowledge of the physiological and molecular mechanisms of phytoremediation began to emerge in recent years, together with biological and engineering strategies designed to optimize and improve phytoremediation. In addition, several field trials confirmed the feasibility of using plants for environmental cleanup. Application: Phytoremediation may be applied wherever the soil or static water environment has become polluted or is suffering ongoing chronic pollution. Examples where phytoremediation has been used successfully include the restoration of abandoned metal mine workings, and sites where polychlorinated biphenyls have been dumped during manufacture, and mitigation of ongoing coal mine discharges, reducing the impact of contaminants in soils, water, or air. Contaminants such as metals, pesticides, solvents, explosives, and crude oil and its derivatives, have been mitigated in phytoremediation projects worldwide. Many plants such as mustard plants, alpine pennycress, hemp, and pigweed have proven to be successful at hyperaccumulating contaminants at toxic waste sites. Over the past 20 years, this technology has become increasingly popular and has been employed at sites with soils contaminated with lead, uranium, and arsenic. While it has the advantage that environmental concerns may be treated in situ, one major disadvantage of phytoremediation is that it requires a long-term commitment, as the process is dependent on a plant's ability to grow and thrive in an environment that is not ideal for normal plant growth. Phytoremediation refers to the natural ability of certain plants called hyperaccumulators to bioaccumulate, degrade, or render harmless contaminants in soils, water, or air. Advantages and limitations. Advantages: the cost of the phytoremediation is lower than that of traditional processes both in situ and ex situ, the plants can be easily monitored, the possibility of the recovery and reuse of valuable metals, it is potentially the least harmful method because it uses naturally occurring organisms and preserves the environment in a more natural state. Limitations: phytoremediation is limited to the surface area and depth occupied by the roots. Slow growth and low biomass require a long-term commitment. With plant-based systems of remediation, it is not possible to completely prevent the leaching of contaminants into the groundwater. The survival of the plants is affected by the toxicity of the contaminated land and the general condition of the soil. Bioaccumulation of contaminants, especially metals, into the plants which then pass into the food chain, from primary level consumers upwards, or requires the safe disposal of the affected plant material. Various phytoremediation processes. A range of processes mediated by plants or algae are useful in treating environmental problems. Phytosequestration, phytochemical complexation in the root zone, reduce the fraction of the contaminant that is bioavailable. Transport protein inhibition on the root membrane, preventing contaminants from entering the plant. Bacola storage in the root cells. Contaminants can be sequestered into the vacuoles of root cells. Phyto extraction, uptake and concentration of substances from the environment into the plant biomass. Phyto Stabilization, reducing the mobility of substances in the environment, for example, by limiting the leaching of substances from the soil. Phyto Transformation, chemical modification of environmental substances as a direct result of plant metabolism, often resulting in their inactivation, degradation, or immobilization. Phytostimulation, enhancement of soil microbial activity for the degradation of contaminants, typically by organisms that associate with roots. This process is also known as rhizosphere degradation. Phytostimulation can also involve aquatic plants supporting active populations of microbial degraders, as in the stimulation of atrazine degradation by hornwort. Phyto Volatilization, removal of substances from soil or water with release into the air, sometimes as a result of Phyto Transformation to more volatile and or less polluting substances. Rhizofiltration, filtering water through a mass of roots to remove toxic substances or excess nutrients. The pollutants remain absorbed in or adsorbed to the roots. Phytoextraction. Phytoextraction uses plants or algae to remove contaminants from soils, sediments or water into harvestable plant biomass. Phytoextraction has been growing rapidly in popularity worldwide for the last 20 years or so. In general, this process has been tried more often for extracting heavy metals than for organics. At the time of disposal, contaminants are typically concentrated in the much smaller volume of the plant matter than in the initially contaminated soil or sediment. Mining with plants, or phytomining, is also being experimented with. The plants absorb contaminants through the root system and store them in the root biomass and or transport them up into the stems and or leaves. A living plant may continue to absorb contaminants until it is harvested. After harvest, a lower level of the contaminant will remain in the soil, so the growth-harvest cycle must usually be repeated through several crops to achieve a significant cleanup. After the process, the cleaned soil can support other vegetation. Advantages. The main advantage of phytoextraction is environmental friendliness. Traditional methods that are used for cleaning up heavy metal-contaminated soil disrupt soil structure and reduce soil productivity, whereas phytoextraction can clean up the soil without causing any kind of harm to soil quality. Another benefit of phytoextraction is that it is less expensive than any other cleanup process. Disadvantages. As this process is controlled by plants, it takes more time than anthropogenic soil cleanup methods. Two versions of phytoextraction: natural hyperaccumulation, where plants naturally take up the contaminants in soil unassisted. Induced or assisted hyperaccumulation, where a conditioning fluid containing a chelator or another agent is added to soil to increase metal solubility or mobilization so that the plants can absorb them more easily. In many cases, natural hyperaccumulators are metallophyte plants that can tolerate and incorporate high levels of toxic metals. Examples of phytoextraction: arsenic, using the sunflower, or the Chinese brake fern, a hyperaccumulator. Chinese brake fern stores arsenic in its leaves. Cadmium, using willow. In 1999, one research experiment performed by Maria Greger and Tommy Landberg suggested willow has a significant potential as a phytoextractor of cadmium, zinc, and copper, as willow has some specific characteristics like high transport capacity of heavy metals from root to shoot and huge amount of biomass production. Can be used also for production of bioenergy in the biomass energy power plant. Cadmium and zinc, using alpine pennycress, a hyperaccumulator of these metals at levels that would be toxic to many plants. On the other hand, the presence of copper seems to impair its growth. Lead, using Indian mustard, ragweed, hemp dogbane, or poplar trees, which sequester lead in their biomass. Salt-tolerant barley and or sugar beets are commonly used for the extraction of sodium chloride to reclaim fields that were previously flooded by seawater. Cesium-137 and strontium-90 were removed from a pond using sunflowers after the Chernobyl accident. Mercury, selenium and organic pollutants such as polychlorinated biphenyls have been removed from soils by transgenic plants containing genes for bacterial enzymes. Phyto Stabilization. Phyto Stabilization focuses on the long-term stabilization and containment of the pollutant. For example, the plant's presence can reduce wind erosion. Or the plant's roots can prevent water erosion, immobilize the pollutants by adsorption or accumulation, and provide a zone around the roots where the pollutant can precipitate and stabilize. Unlike phytoextraction, phytostabilization focuses mainly on sequestering pollutants in soil near the roots but not in plant tissues. Pollutants become less bioavailable, and livestock, wildlife, and human exposure is reduced. An example application of this sort is using a vegetative cap to stabilize and contain mine tailings. Phyto Transformation. In the case of organic pollutants, such as pesticides, explosives, solvents, industrial chemicals, and other xenobiotic substances, certain plants, such as cannas, render these substances non-toxic by their metabolism. In other cases, microorganisms living in association with plant roots may metabolize these substances in soil or water. These complex and recalcitrant compounds cannot be broken down to basic molecules by plant molecules, and, hence, the term phytotransformation represents a change in chemical structure without complete breakdown of the compound. The term green liver is used to describe phytotransformation, as plants behave analogously to the human liver when dealing with these xenobiotic compounds. After uptake of the xenobiotics, plant enzymes increase the polarity of the xenobiotics by adding functional groups such as hydroxyl groups. This is known as phasometabolism, similar to the way that the human liver increases the polarity of drugs and foreign compounds. Whereas in the human liver enzymes such as cytochrome P450S are responsible for the initial reactions, in plants enzymes such as peroxidases, phenyloxidases, esterases and nitroreductases carry out the same role. In the second stage of phytotransformation, known as phase 2 metabolism, plant biomolecules such as glucose and amino acids are added to the polarized xenobiotic to further increase the polarity. This is again similar to the processes occurring in the human liver where glucuronidation and glutathione addition reactions occur on reactive centers of the xenobiotic. Phase I and II reactions serve to increase the polarity and reduce the toxicity of the compounds, although many exceptions to the rule are seen. The increased polarity also allows for easy transport of the xenobiotic along aqueous channels. In the final stage of phytotransformation, a sequestration of the xenobiotic occurs within the plant. The xenobiotics polymerize in a lignin-like manner and develop a complex structure that is sequestered in the plant. This ensures that the xenobiotic is safely stored, and does not affect the functioning of the plant. However, preliminary studies have shown that these plants can be toxic to small animals, and, hence, plants involved in phytotransformation may need to be maintained in a closed enclosure. Hence, the plants reduce toxicity and sequester the xenobiotics in phytotransformation. Trinitrotoluene phytotransformation has been extensively researched and a transformation pathway has been proposed. Role of genetics, breeding programs and genetic engineering are powerful methods for enhancing natural phytoremediation capabilities, or for introducing new capabilities into plants. Genes for phytoremediation may originate from a microorganism or may be transferred from one plant to another variety better adapted to the environmental conditions at the cleanup site. For example, genes encoding a nitroductase from a bacterium were inserted into tobacco and showed faster removal of TNT and enhanced resistance to the toxic effects of TNT. Researchers have also discovered a mechanism in plants that allows them to grow even when the pollution concentration in the soil is lethal for non-treated plants. Some natural, biodegradable compounds, such as exogenous polyamines, allow the plants to tolerate concentrations of pollutants 500 times higher than untreated plants, and to absorb more pollutants. Hyperaccumulators and biotic interactions. A plant is said to be a hyperaccumulator if it can concentrate the pollutants in a minimum percentage which varies according to the pollutant involved. This capacity for accumulation is due to hypertolerance, or phytotolerance, the result of adaptative evolution from the plants to hostile environments through many generations. A number of interactions may be affected by metal hyperaccumulation, including protection, interferences with neighbor plants of different species, mutualism, commensalism, and biofilm. Table of hyperaccumulators hyperaccumulators table Euro 1, Al, Ag, As, B, Cr, Cu, Mn, Hg, Mo, naphthalene, Pb, Pd, Pt, Se, Zn, hyperaccumulators table Euro 2, Nickel, hyperaccumulators table Euro 3, radionuclides, hydrocarbons, organic solvents. Phyto Screening, as plants are able to translocate and accumulate particular types of contaminants, plants can be used as biosensors of subsurface contamination, thereby allowing investigators to quickly delineate contaminant plumes. Chlorinated solvents, such as trichloroethylene, have been observed in tree trunks at concentrations related to groundwater concentrations. To ease field implementation of phyto screening, standard methods have been developed to extract a section of the tree trunk for later laboratory analysis, often by using an increment borer. Phyto screening may lead to more optimized site investigations and reduce contaminated site cleanup costs. See also, phytoremediation plants, phytotreatment, biodegradation, remediation, bioaugmentation. References. Bibliography, a OE Phytoremediation website. A includes reviews, conference announcements, lists of companies doing phytoremediation, and bibliographies. A OE an overview of phytoremediation of lead and mercury. June 6, 2000. The Hazardous Waste Cleanup Information website. A OE enhanced phyto extraction of arsenic from contaminated soil using sunflower. September 22, 2004. U.S. Environmental Protection Agency. A OE phyto extraction. February 2000. Brookhaven National Laboratory 2000. A OE phyto extraction of metals from contaminated soil. April 18, 2001. Lassett, July 2002. Donald Bren School of Environment Science and Management. A OE Phytoremediation. October 1997. Department of Civil Environmental Engineering. A OE Phytoremediation. June 2001, Todd Zender. A OE Phytoremediation of lead in residential soils in Dorchester, Moore. May, 2002. Amy Donovan Palmer, Boston Public Health Commission. A OE Technology Profile, Phyto Extraction. €1,997. Environmental Business Association. Vassal A.D., Kapulnik Y., Raskin I., Salt D.E., The Role of EDTA in Lead Transport and Accumulation by Indian Mustard, Plant Physil 117, 447. A 53, doi, 10.1104/pp 117.2.447, PMC 34964, PMID 9625697. Salt, D.E. Smith, R.D. Raskin, I. Phytoremediation. Annual Review of Plant Physiology and Plant Molecular Biology 49, 643. A 668 doi, 10.1146/Annuary Plant 49.1.643. PMID 15012249. Ko, T. Lee, H. Y. Cheng, Y. Zai, and S. Yun Mokai. A OE Development of Profitable Phytoremediation of Contaminated Soils with Biofuel Crops, a Journal of Environmental Protection, Volume 4, pages 58 to 64, 2013. X. J. Wang, F. Y. Lee, M. Okazaki, and M. Sujzaki, a OE Phytoremediation of Contaminated Soil. Annual Reports S, Volume 3, pages 114 to 123, 2003. External links: Missouri Botanical Garden, Phytoremediation website. Review articles, conferences, Phytoremediation links, research sponsors, books and journals, and recent research. International Journal of Phytoremediation. Devoted to the publication of current laboratory and field research describing the use of plant systems to remediate contaminated environments. Using plants to clean up soils. From Agricultural Research Magazine, New Alchemy Institute. Co-founded by John Todd.